My name is Ross Maddox, and I'm a postdoctoral researcher at the University of Washington Institute for Learning and Brain Sciences. I'm a Hearing Health Foundation awardee, and my work is supported by the Royal Archmasons International. In the next few minutes, I'll tell you about my work and how I hope it will help people with Central Auditory Processing Disorder, or CAPD. CAPD is when you have supposedly normal hearing, which is what an audiologist would measure, yet you still have trouble listening in a lot of situations.
In your left ear, raise your left hand, and when you hear a tone...
And what the central means is essentially after your ears. So most hearing issues that people talk about tend to be somewhere in your middle ear or your inner ear, also known as the cochlea. After the cochlea, sound gets turned into neural impulses in your brain. And with CAPD, there's some disconnect there that makes it really hard for people with well-functioning ears to listen. Let's say you're in your favorite restaurant. There are lots of people talking, maybe some music playing, and your friend's telling you about their recent vacation. The brain is remarkable in its ability to focus on one sound and ignore many others. This is what listening at the restaurant sounds like to most people. You have to work a little harder, but you can still understand me. But for someone with CAPD, it will probably sound more like this. Pretty difficult. With CAPD, you can't separate your friend talking from the rest of the noise. The focus of my work is actually how people with typical abilities and also people with CAPD use visual cues when they're listening. So you may not even know you're doing it, but when you're in a loud place talking, you're probably watching the person's mouth as they speak, and this gives you a lot of important information. We know that this helps people with hearing issues that, say, might use a hearing aid or people with cochlear implants, but we really don't know if people with CAPD are able to derive the same benefit, and if not, is this one of the causes for why they struggle? Let's do a demonstration. We're going to bring in some background noise, and I'm going to show you a video of someone speaking. I'm going to show it to you twice, once with their mouth covered, and then again with their mouth visible. Your job is to try and listen to what sentence the person is saying.
The cool blue creek runs through the deep green wood.
Now here it is with their mouth visible.
The cool blue creek runs through the deep green wood.
Most people find the second example to be much easier because they're able to use the visual information to help them listen. So the types of experiments we do are twofold: behavioral and neuroimaging.
Right. And I'll clip this on your left collar.
In the behavioral experiments, we design very specific types of audio and video, play them for volunteer subjects, and analyze their responses. We can do the same task for people with CAPD and without, and compare their performance. This tells us about how the two groups use visual cues differently when listening. The second type of experiment we do is called neuroimaging. We use special equipment to measure the minuscule electrical currents that cells in the brain use to communicate. Specifically, we use things called electroencephalography, or EEG, and magnetoencephalography, or MEG. And what these do is they let us measure the fields outside your head that are created by all the neural activity going on inside. We can then figure out which areas of the brain are active at what time and relate this to how well people are listening. The first benefit of this work is that it'll help separate different populations within the CAPD class that might be having different problems, yet they have the same diagnosis. The second related benefit is that by better understanding what's happening in the brain, we'll also be able to design better treatments that may be more catered to a certain person's difficulties. We so far have completed one behavioral experiment and one neuroimaging experiment. Both of those have been presented at an international conference for studying hearing. Now that data collection is done, I'm also in the process of submitting to scientific journals for publication. I'd really like to thank the Royal Archmasons International and the Hearing Health Foundation for supporting this work.
name is Edward Bartlett. I'm an associate professor in biological sciences and biomedical engineering at Purdue University. A major part of my research in my lab has been generously funded by the Hearing Health Foundation and by the Royal Arch Masons. The work described here was carried out mainly by two graduate students, Yamini Venkataraman and Kal Rabang, and they would have not been able to complete their projects or their PhDs without the support of the Royal Arch Masons. My research focuses on the development of neurons in the rat auditory thalamus. The auditory thalamus is the information bottleneck of our hearing system. Nearly all auditory or hearing inputs to the cerebral cortex must pass through the auditory thalamus. Despite this, little is known about how the auditory thalamus changes during development. Our early environment has the potential to influence the maturation of our hearing abilities and can potentially have lasting consequences. It is already known that developing hearing systems are quite sensitive to their surrounding environments, which could include noisy urban environments or hearing loss due to persistent ear infections. One critical aspect of hearing is the ability to accurately represent rapid changes in sound amplitude, meaning sound level, or sound pitch, an aspect that is known as temporal auditory processing. Reduced temporal auditory processing could lead to difficulties in sound recognition and speech comprehension, such as what has been observed for Central Auditory Processing Disorder, or CAPD. Sounds are encoded by patterns of signaling between neurons in our brain. My work investigates how that signaling develops in the auditory thalamus of the rat. Rats and other rodents are very useful for studying brain circuits in hearing, because rats don't begin hearing until about 11 to 12 days after they've been born, unlike human infants who can hear well before they're born. Therefore, for rats, it's possible to study neurons just before and just after they have experienced processing sounds. Our preliminary studies were in typically developing animals, providing a baseline to use for future studies with animals from altered sound surroundings such as hearing loss or ongoing background noise. For these initial studies, we wanted to figure out what determined the, quote, speed limit for these neurons in the auditory thalamus. Whether it was the speed limits of inputs to the neurons themselves, meaning what sensitivities they've inherited, the connections between the neurons, called synapses, or speed limits imposed by the neurons themselves. In order to test these possibilities, we employed two main techniques. We recorded responses for large populations of neurons that are inputs to the auditory thalamus. We tested the ability of these neural populations to follow sounds that were either slowly or rapidly changing in sound level or sound amplitude. We found that although the responses to sound were larger in older rats, both older rats and rats that had just begun hearing were able to follow the sound signal even at relatively rapid frequencies. So that was only a bit different. This suggested that the inputs to the auditory thalamus were not the main limiters of temporal processing. These measurements, called auditory evoked potentials, are quite useful because nearly identical measurements may be obtained in human infants and children. Next, we tested the synaptic connections between neurons by gently rupturing a cell with a small glass recording electrode that enabled us to record small electrical changes in individual neurons using a technique called whole cell patch clamp recording. From these measurements, we determined that while the connections between the neurons got stronger with age, they were not getting faster, so the speed limits weren't really changing. However, it turned out that the main limiting factor limiting the speed and precision was the neurons themselves. The neurons became much faster and more able to handle rapid inputs over the first 3 weeks of a rat's life, which would correspond to a juvenile stage in humans going up to about 10 years old or so, especially comparing just before and just after hearing begins in rats. Now, this work has been presented at national conferences and has resulted in multiple publications in high-quality journals, such as the Journal of Neurophysiology. It will form the basis of an upcoming grant to NIH, an R01 submission, that will include it as a motivating component, looking at potential correlates of Central Auditory Processing Disorder, meaning CAPD. My sincere thanks to the Hearing Health Foundation and to the Royal Arch Masons for driving the research, for promoting the scientific development of 2 students, and for supporting work which may lead to the generation of a viable animal model for CAPD, which currently does not exist. Thank you.
Hello, I'm Dr. Elizabeth Dinsis, a physician scientist at the Albert Einstein College of Medicine in Bronx, New York. My research focuses on how our brains make sense of our noisy world. Working with Dr. Elise Sussman in the Cognitive Neuroscience Laboratory, our emerging research grant project studies how the brain separates out the sounds we want to hear from the background noise, like we do when we want to listen to one person's story in a noisy social gathering. Normally, people are able to focus on a particular person's voice and easily understand what they are saying. But people with Central Auditory Processing Disorder, or CAPD, can struggle to understand what they hear. With funding from the Royal Arch Masons through the Hearing Health Foundation, we are researching what deficits contribute to CAPD, looking at the abilities to automatically distinguish between similar sounds and to focus and maintain attention within a background of many sounds. Our emerging research grant project looked at the brain patterns and performance of normal hearing elderly listeners who have developed difficulties in following conversations in noise as a model for CAPD in all ages. Using brainwave recordings and listening performance, we analyzed their responses in environments with varying degrees of listening difficulty and looked at the brain processes during both passive listening and active discrimination between similar sounds. Our preliminary results have identified impairments in both automatic sound discrimination and in focusing attention on target sounds in competing noise. Our ongoing research will further characterize the specific factors and types of brain processing problems that are contributing to CAPD. We will identify those processes in the brain that can help people with CAPD pick out and correctly identify sounds to become successful listeners in noise. The long-term benefits will be an understanding of the basis for CAPD and will provide a foundation for improving listening strategies and treatments to help children and adults with CAPD better understand speech in noisy situations. The grant we received has led to presentations at the Society for Neuroscience and at the combined otolaryngological spring meetings. Your support has also led to the preparation of research manuscripts for publication and the submission of a grant proposal to the NIH. We are enormously grateful to the Royal Arch Masons for their interest in our research and for their generous support, which has provided us the opportunity to continue this important work and to contribute to bettering the lives of individuals with CAPD. Thank you.
Hi, my name is Alan, and I'm currently a research associate at the University of Wisconsin-Madison. Much of my research work is with those who have bilateral cochlear implants, A cochlear implant is a medical device implanted into the ears of those who are profoundly deaf, and it has the ability to restore speech understanding, especially in quiet situations. However, in noisy situations, people with implants still struggle to hear well, and I've been working on strategies to improve this situation. Most bilateral implant patients report having a better ear for listening, and this led to the idea of a better ear listening strategy which will help them to hear in noisy environments. Imagine the scenario where you're struggling to have a conversation with your wife in a noisy environment. The concept behind the better ear strategy is to apply smart engineering that would separate out the voice of your wife from the surrounding background. Send the voice of your wife to your better ear so that you can hear her better, and the remaining sound scene to your other ear so that you can still know what's going on around you. The idea is that you switch your attention between your better ear and your other ear so that you can decide on what you want to listen to. With the money that we've been receiving from the Hearing Health Foundation, we've been testing this idea with bilateral cochlear implant patients, and we've been seeing some great results. I believe the better ear strategy can be applied to those with CAPD to help them hear in noisy environments. One key question is trying to figure out which is the better ear for those with CAPD. And I am teaming up with Dr. Matt Wynn, who has been developing new tests to measure listening effort. Once we have a reliable measure which is the better ear, we can then apply the better ear strategy to those with CAPD. I'm very thankful for the support I've received from the Royal Archmasons, and I hope you are excited as I am about the possibilities of this strategy to help those with bilateral cochlear implants, CAPD, and other forms of hearing impairment hear better in noisy environments. Thank you.